Welcome to the biology session. Now we are going to see about the artificial methods. In the previous two videos, I uploaded asexual reproduction in the natural vegetative method. Now we are going to see in the vegetative reproduction in the artificial meaning. Okay. So apart from the above mentioned natural method, that means I uploaded two video for that natural vegetative methods. So now we are going to see the number of methods that human used for agriculture and horticulture, these methods, that is called artificial methods. So many number of methods man followed in horticulture and agriculture. Such methods are said artificial propagation. Propagation means nothing but it is a cultivation. And some of the artificial propagation methods have been used by man for long time, means that is called conventional methods. It's a age old methods that also we are have to follow now also. And nowadays so many modern sophisticated techniques are now followed in the agriculture and horticulture to propagate plants into a very short time with very large plants. For example, tissue culture, likewise gene cloning, likewise. So these methods are called modern method. So artificial methods divided into two types. One is conventional method, another one is the modern method. Now we are going to see about the conventional method. So the conventional method, now we are going to see this is divided into cutting, grafting and layering. So these three methods are used by farmers. First one is the cutting. It is nothing but simply telling cutting means we take the plant pots by cut and we are placed it into the soil, means it will be growing. So it is a very simple method. There are three types are there in the cutting. One is the root cutting. Some plants produced by root cutting, that is one of the example is malus plant. So in this just we cut the malus plant root and we are placed into a pot or any soil, means it will be growing into a new plant. Likewise the stem cutting. For the stem cutting, stems we are cutting from the plant by cutting the branch and we are placed into the medium. The medium we are suitable medium we are giving to that plant pots, that is called soil. So if you are placed into the soil, means it will be growing into a new plant. Stem cutting example hibiscus, bougainvillea and moringa. That means in Tamil it is called sembaruthi, kagidappu and murungai maram. And leaf cutting, begonia and brabellum, two special plants are growing by this leaf cutting. And among these three cuttings, stem cutting only used in large manner. That is this stem cutting only widely used propagation techniques among these three. And we know that the cutting is named by what portion we are prefer for the cutting. So for example, we are prefer stem for stem cutting, means the method is called stem cutting. Likewise the name is given. If you are taking the root for cutting, means it is called root cutting. If you are taking leaf, means it is leaf cutting. The second one process is called grafting. This technique is employed by taking two plants and we are placing these two plants and mingled with each other and allowing to grow into a single plant. That is called grafting. For the grafting technique, we use two plants. One plant called stalk, another plant called scion. The stalk means nothing but we growing one plant in the soil itself, and one plant only we are taking a branch only from the plant. So one plant is rooted, remain in the soil, and another one plant we take the branch to make the graft. That's all. So one plant that always contacted into the soil, means that plant is called a stalk. Another one plant we are taking the branch only, means that is called a scion. So example, citrus, mango and apple. These three plants are nowadays growing by these grafting techniques. And 5 types of grafting are there. One is bud grafting, approach grafting, tongue grafting, crown grafting and wedge grafting. These 5 types are mentioned based on the method of uniting the scion and the stalk. How the stalk is united with the scion. So based on these techniques, 5 types are nowadays followed. First one, we are going to see the bud grafting. Here we take the stalk. Stalk means one plant we are growing in the soil, remain in the soil. It's not, that is called stalk. From the stalk we take the stem portion and we are made a one T-shaped cut and elevate the bark. Then one gap will be formed and we take which plant we are going to graft, means that is called scion. From the scion plant one small little wood is taken and this wood is inserted into the T-shaped incision in the stalk region and tightly bandaged with a tape. This is called bud grafting. In the approach grafting, in this method both the scion as well as the stalk remain rooted. That means both the plants are allowing to growing in the soil, but one plant we growing in the pot region, another one naturally growing in the soil. That naturally growing plant in the soil we cannot move, but we can move the pot plant, bringing together into the scion plant. So when we take or bring the stalk plant near to the scion plant, then we prefer the two branches from the two plants with the same size and made a small slice, that means a small cut, and these cut portions are now joined closed with each other and held by a tape. So after 1 to 4 weeks the tip of the stalk and the base of the scion are cut off. At the time the branches will produce leaves so that they are growing and detached and growing into a separate pot as a new plant. In the tongue grafting, a scion and a stalk having the same thickness also taking, and they are cut obliquely, that means crossly they are cut, and they are the scion and the stalk plants are fit into each other and which are tightly bound, that means tightly joined with a tape. In the crown grafting, the stalk is taken as a large size, and scions are cut into wedge shape. That means wedge, I think you often see this wedge shape, that is crossly made a cut and sharply made a cut in the scion, and we are taking the scion usually in the very slender manner, and it will be insisted into the cut that made in the stalk region, and it will be fit with the cleft or slits that made in the stalk region, and it will be bound with a tape or a graft wax. The wedge grafting, in this method a slit is made in the stalk or the bark is cut. So the bark region of the stalk region stem is only making to cut, and a twig, that means a small branch from the scion plant is inserted and tightly bound so that the cambium of the two is joined. Cambium is nothing but the cambium is a special type of tissues that responsible for producing the xylem and phloem, and which is present in the bark region. So we are made the cut in the stalk and scion at the bark region only, so that two barks are now mixed together, means the cambium of the two segments are joined together and allowing them into a new plant. So third one, conventional method is layering. Here in this method the stem of a parent plant is allowed to develop roots while still intact. So what they are telling is some plants produce a flexible stem portion, means we can easily bend that portions and we can bury partially some portions into the soil, means it will be produce some roots, so that after the roots are produced, the portion, that means the branch is cut from the mother plant and started to growing as a new plant. That is called a layering. There are two types of layerings there. One is a mound layering, another one is a air layering. Example, jasminum and ixora plants. In the mound layering, this method is applied for the plants having flexible branches. So the lower branch, the very lower branch with the leaves are just bent to the ground, and they are having part of the stem is buried in the soil, and the tip of the branch is exposed above the soil. So after the roots emerge means it will be easily cut from the mother plant and it started to growing as a new plant. That's all. In the air layering technique, the stem is girdled at a nodal region. Nodal means it is nothing but the point of attachment of leaf into the stem. That portion is called a nodal region. In this nodal region, the bark is girdled. That means the bark is completely removed around the stem. That is called a girdle. So in this girdled region, we apply some root inducing hormone and then moist or damp soil, and we are packaged it using a polythene sheet. Means after sometimes the root will emerge. After the root emerge means we can easily cut the branch and shifted it into the new pot and allowing them to grow into a new plant. This is called the air layering method. So in the first diagram you can see that the lowest branch, flexible branch is partially buried into the soil, and afterwards the root is producing means it will be cut from the mother plant. The second one diagram shows the air layering technique. But in the air layering technique, in the second diagram we are choosing the branch not from the bottom side. So we are choosing the branch at the tip, and we are making the girdle and take or remove the bark region, and then it will be applied with the hormone, that is a root inducing hormone, and mud soil, and it will be tightly bandaged with a polythene sheet. So it will be produced root means it will be cut from the mother plant, and the branch is allowing to grow into a new plant in a separate pot. So this diagram shows the grafting. First one is a bud grafting. Here in the stalk plant branch, a T-shaped incision is made with a knife, and then the bark is elevated. In this gap, the scion branch, a little wood is taken from the scion plant, and it is placed into this gap and tightly bandaged with a tape. The second one is the approach grafting. Here both the scion as well as the stalk, both the plants are taken, remain as rooted in the soil, and the branches we are preferring is similar in size from the two plants that you can easily see in the diagram, and it will be make a small slice cut and it will be joined together with a tape. Here you can see the tongue grafting from the diagram. We are taking the stalk as well as scion and made a cut as a oblique cut. This is a oblique cut you can see here in the diagram, and we are properly fit the scion into the stalk region by a graft wax. And another one is a crown grafting. Here the stalk is very much large in size when compared to the scion. The scion is very much smaller in size, and make a cut and it is fit into the layer of the stalk region at the cambium region. In the diagram you can easily see this, the cambium is present in the bark, so that the bark is cut and it will be fixed with the scion branch. The last one is the wedge grafting. Here the stalk as well as the scion both are having a similar in size, and you can notice the wedge shape cutting in the scion branch, and it will be placed or insert into the stalk and properly bandaged. So now we are going to see the advantages of conventional methods. So in the conventional methods, the plants produced are genetically identical because we don't use the male as well as a female plant. We are using only one plant, so that they are genetically identical, and many plants can be produced quickly. So for example, we don't wait for the plant to produce a flower, fruits and seeds. Just we are taking the branches and we are cultivating the plants. So it is a quick method. Third one, some plants are produced little seed, or some plants are produced no seeds, means we are using this technique. And fourth one, some plants can be propagated more economically. So more commercial plants are also grown by this technique. Example, Solanum tuberosum, that is a potato. And if you are preferring two quality of two different plant, we can blend with two quality into one plant, means it is possible with the help of the grafting techniques. Say for example, if one plant is noticed for high yielding variety, means that is a dominant quality. So this quality and another one plant having a disease resistant quality, means we can easily blend with these two qualities into one plant by using the graft technique. So now we are seeing the disadvantages of conventional methods. So use of virus infected plants as parents, means this will produce a viral infected new plants. So that we are preferring for these techniques, conventional techniques as parents, means that plants should be free of any disease. That is very much important. And for the next one, vegetative structures used for propagation or bulky. Say for example, the stem roots are very much larger in size, so that it is very much difficult to handle and store these things. So thanks for watching. Please subscribe. Thank you.